Hello, everyone. My name is Jimmy Lin. I'm with the Rare Genomics Institute. And today we are talking about a topic that actually is very pertinent to the conference here. It's beyond genomics, preparing for the avalanche of post-genomic clinical findings. I think a lot of the talks here talk about genome sequencing and starting to do it for patients. The question really is, what's the next step to be able to help these patients? A lot of these patients would have they find a new gene or they have a list of genes. The question is, how can we sort of continue to help them? So that's a question that we at Rare Genomics are trying to do. And hopefully we'll dig a little bit into this problem and then some of the solutions that we're preparing, and that together, I think as a community, we can start thinking about this greater problem. We know that it's, we're a small group, and it's not going to be us who's going to be solving it, as the whole community is going to do it together. So a little bit about our background. We are a nonprofit that is mostly in the US, but also we have sites in Singapore, Malaysia, Australia, Israel, now in China, South Korea that are coming. And what we do is we connect rare disease patients with clinical sequencing sites and help them with the logistics, finding the right clinician, finding the right clinical geneticist, sequencing capacity, helping interpret the information if they need it, and then also sort of helping them with fundraising if their insurance is not able to sort of cover that. As many of you know, is that there are millions and millions of patients that have rare diseases that are undiagnosed, and many people in this conference know about the whole diagnostic odyssey with rare disease patients. And this is a very large problem, and be able to solve it does require not only sort of top-down efforts, but as well as bottom-up efforts, which we're trying to do from empowering and enabling patients themselves, partnering with academic institutions and commercial ventures to be able to work together. And this number that people throw out, between 7,000 and 8,000 rare diseases, and each of them, there's probably going to be more as more genome sequencing is done, and all these large initiatives like the Mendelian Centers or initiatives in the UK. And this, as a long tail problem, is a little bit different than what science is used to in terms of a top-down system, and that's what we're trying to sort of build from the bottom up. And we know that rare disease, ultimately, even if you diagnose, therapies are going to be what's most important. And currently, less than about 3% of rare diseases have any type of therapy and no cures. So as people who work in genomics, our goal really is to be able to help the patients. So diagnosis is really the first step of this process. So this is what we at Rare Genomics is trying to do. So I'm going to just talk about quickly what we do in the genomic side, and then we'll be able to sort of dive deeper into sort of the post-genomic side. And this part, would be a little bit quick. So what we do is to provide three separate aspects of this: technology, scientists, and funding to this. And a lot of this conference, you will be the scientists and technology that would be needed for this. And some of the stuff that we've done would sort of tailor and would actually fit with a lot of what people are doing in this conference. So one of our first cases is about sort of 3 years ago now, which seems like eons ago in genomics. Is one of our first patients, Robert. He's one of your classic diagnostic odyssey patients. He was born healthy, and about at age 1, he had a neurological decline, unable to use his muscles, unable to feed, rushed to the hospital, and started their whole diagnostic odyssey process. By the time that they came to us, dozens of years later, around 14 years later, I believe, this was at the beginning of the whole genomics era. Nobody had really even figured out what kind of disease they had, whether it was a mitochondrial disease, whether it's a genomic disease. So we set off starting to do that. And this is before all sort of the next-gen sequencing is now sort of prevalent for genetic diseases. So the thing that we did in the past and we continue to do is connect them with the right technology, of which right now mostly what we do is sort of NGS type of sequencing with exome capture, and that's pretty standard. And if you're at this conference, you probably sort of know a lot about this. The second thing that we do is then we connect them to either commercial sites or research studies that will be able to help. These are some of the study centers that we've participated in the past that have either created, whether they have already commercial pipelines to do that, whether they have in research pipelines, It all depends on whether they fit into certain research criteria, whether they have CAP-CLIA services that's ready for it that we can connect them to. And now we're starting working a lot with sort of overseas, which don't have the luxury of all these sort of clinical and NGS services. And like I mentioned, we're in India, Singapore, Malaysia, Australia, and working with partners all over the world. In the U.S. is, the clinical services are much more available now. The hard part is still actually insurance reimbursement. So that's where really the third step comes in. I mean, the beginning when we were doing this, there was basically no reimbursement whatsoever for this. So we created a platform where patients can raise funds through their friends and family. And now, although there's some of our patients are getting reimbursement, but it's still a hard road for many patients. And so we help them and connect them with using our pipeline. So we not only have a pipeline flow, we have coaching to help them. We have template letters that can be sent out. So our goal is with these patients, if they don't have the right technology, we connect them to the right centers. If they have the right centers, we can then help them with fundraising. Really, as a nonprofit, we help the patients from beginning to end. So this is sort of from our old screen capture site. Some of our patients that we will create a page for them and most people know on this conference is it costs about less than $10,000 and to be raised, and we sort of help patients be able to raise this. It can happen really depending on the social network of the individual. We've done campaigns as quick as 1 day, which is actually crazy to sort of see the money come in, where the average is a few months for people to be able to raise funds. Once the funds are raised, then we release all the funds then to the funding minus processing fees from the credit card companies, and then but we don't sort of take any money from that. It goes directly to the patient to be able to help that. And with sort of up and down of reimbursement environment, I think this is going to be an important aspect that we will continue to play. So what we like to think about is this picture. This obviously is not the actual team, but we think about for Robert going from being someone on a diagnostic odyssey to now not only scientists be able to help them, but funders and researchers all over the world to be able to help. And like many other diagnostic odyssey stories, we were able actually to find the gene from him, and his gene is PRKA, and it's a type of dystonia called dystonia 16. He is one of less than approximately a dozen patients in the world that has this, and we're excited. But then the question is, what next? You found the gene, but how can we really be able to leverage that to be able to help the families? So we've partnered and in discussions, and these are sort of evolving with and with other groups, and these are many other groups that do this, right? Now there's a large really, whether it was Forge or Center Mendelian Genomics, there's a big avalanche of data that's coming out. But then the question is, what's next going to be help them after you find the gene? What really is helpful about that gene? So we really think diagnosis is that first step, and the real work is at the sort of the bottom of the iceberg, if you will. And this is really the avalanche of data. So in the beginning, when you have those initial stories of ending diagnostic odysseys, like Nick Foker, or for example with the Berry twins, right? You have a lot of scientists really able to follow up. But as we have more and more of this data coming out, it is no longer just simple to be able to find scientists who will be able to take these studies and start doing functional studies to be able to understand these genes. Many of these genes are unannotated. So then the question is, how do we do that? The number that within the field that people report is approximately a quarter to a third of these diagnostic odysseys end in some sort of result. But then the question is, what do you do with the test? And for us, what we really think that this is sort of not sort of like a Rosetta Stone, written in stone kind of process. This really needs to be a living document, if you will. Because as annotation databases get better, as more and more genomes are sequenced, actually initial quote unquote findings that are high suspects of genes for these patients often will turn out not to be. So how do we go from sort of looking at it as a sort of static process to something that is much more sort of dynamic and fluid? So the way to do, so what we think about really is more of the process where there's constant editing, and this really needs to be more than a report that is a static piece of paper that's integrated into the patient record, but ways that we can continue to go back and relook at and annotate data. And this is going to be very hard, right? This is not going to be an easy task to continue to go back and look at the data, reanalyze it, and as more and more patients have this data, then and how are we able to be able to help? And as we know that, right, even in every step of the process, whether it's generating bases or sequence alignment or variant calling, there's better knowledge bases, there are better calling algorithms, calling not just based on one sample, but on multiple samples, better alignment algorithms, and it'll keep on getting better. So then how can these patients be able to really benefit from that? So we see this process really we really are in the larval stage with helping these patients in the very, very beginning of this, and how can we build an ecosystem that we can really think about it as it goes to the next step. So what we are proposing is truly build an ecosystem for that. It's just a picture, and it's going to require many, many different components of that, and we're at the beginning of doing that, and we'll talk to you a little bit overview of what we're trying to build in terms of each of these components at RGI. So these components really come from three aspects. First, storage and sharing. You need a place to store all this data that patients can access. Number two is scientific validation, to really find is the gene candidate really that gene? And then lastly is connecting patients to each other, connecting to scientists to really understand that more. So let's first talk about storage and sharing. And this is going to be a little bit different, and every context is going to be very specific in terms of return of data. How much should be returned? Is it a piece of paper? Is it one gene? And this continually evolve in this discussion here. But what we're hoping and what we're trying to do is, if we can give the patient the most data that they can have access to with sort of guidance and expertise, they potentially be able to sort of make more use of this data and have this as a sort of a living document. So here we have systems to be able to store three types of data. The standard medical record data, where it's often very important in terms of be able to understand the genome. The research data. So again, this is sort of people. You could be as radical as the PGP to return everything. You could return the VCF files and how patients are going to work with that. It's constantly sort of debated. But what we're hoping to do is, we're partnering with so that we can be able to return research data, actually VCF files or BAM files, even back to the patient. And then lastly, be able to store actually biospecimen and biobank in case they want to do any type of follow-up work that already it's a biobank that is their own specimen, and that if you want to do additional analysis like transcriptomics, proteomics, that you have that ready. So we already have some partners we're in discussion with, or further discussion. Patients Know Best is what we use for right now storing all our electronic medical records. That's sort of HIPAA compliant. We're working with and continually working with the Open Medicine Institute, be able to store VCF data. Some of our VCF data also we can store with Patient Know Best, which actually is now working with to Genomics. So then they can put additional ANNOVAR annotation on that as well. So we see this as a trend. It's not going to be an easy transition, and people are not used to sort of what does it mean if we give all this data back. But I think there's ways that we can start to sort of have this conversation. And lastly, working with groups like the Genetic Alliance to be able to store biobanking in a patient-specific way. You don't just donate it to a university, to an advocacy group, but they themselves get to have ownership of that data. It's controlled by the patient. So with this, you really can then store your records, your data, your specimen, of which then you can start thinking about how you can act upon it to be able to make the most of this information, whether you have a gene of interest, list of genes of interest, or you're one of the 2/3 that you don't know what to do next, and with additional annotation, you hope to be able to find things for them. So the second sort of leg of this process is sort of scientific validation. And what we do is there's, again, three aspects that we do. Firstly is sort of crowdsourcing ideas. And I'll sort of talk a little bit more about that, is how can we sort of leverage all the different scientific expertise in the world potentially to be able to weigh in on this, because rare diseases span the gamut of biological study. There's there's not ever a large enough group of experts to be able to help them. Secondly, is computational reanalysis to run different pipelines to be able to do that. And we're in discussion with different groups to be able to do that. And then lastly, experimental validation. So let me talk about the first thing, crowdsourcing of ideas. We're doing that right now. We're just sort of piloting in-house and just sort of simple bulletin board system where we, with the patient's permission, be able to post anonymized sort of information about the case, and scientists will then be able to sort of weigh in on potential strategies, whether additional molecular profiling is needed, whether they're an expert in this gene, other experts. We also connect them to people working on those genes, through PubMed and others, and really to be able to find the best experts to be able to help these patients. The second thing, if we sort of go back to sort of the 3 stages, computational reanalysis. And these are some of others that we've sort of in discussion and worked with in the past, who have different pipelines that we can rerun. And these are just sort of representative. But as better algorithms come out, as better annotations database come out, I think being able to really analyze and really be able to give patients the ability to do that. And of course, there's a lot of caveats, with what's happening with 23andMe, what's happening with what does it mean to be sort of direct to consumer. But with sort of the proper protocol, with IRB oversight, there are ways that we think that the patient can be really empowered and engaged in this process, and for that reanalysis to occur, not necessarily have all the burden on the companies who sort of do it initially. But this is something that we're piloting ourselves to sort of think about to build this, and we think that this could potentially, this really is needed because the data we're getting in right now is still so sparse. With better databases, this reanalysis we think is going to be very, very important. The last part of this sort of scientific validation process, as I here, is experimental validation. So experimental validation is obviously very, very difficult for the patient to do when we're not suggesting sort of a DIY bio movement, but more we partner with CROs to potentially do this. So our main partner is Assay Depot, which is our CRO marketplace that has databases of thousands of different CRO companies. So then if the crowdsource of scientific ideas can propose a certain study, maybe that they can, the patient themselves can, in the same way that they fund some of the genomic studies, fund additional studies or creation of reagents to be able to help understand and take it to the next stage. And we've done this not only on an individual basis, but also with as a large competition. So in the past, these are some of the companies that we've partnered with to give away either software or reagents or even sort of monoorganisms to be able to understand individual cases. And there are many instances now in the literature, whether it's the mite family or the Wowsy family, whether the Beery family, the Hempels, a lot of families are pushing forward research on their own. But it's a large heroic effort and takes really special infrastructure and family members to be able to do that. And what we want to do is make that much easier and create this partnership. And ultimately, our goal really is, we don't think that we're these simple systems and the ecosystem that we create would be able to solve it. And at the end, it's connecting to the right groups and researchers to really take that to the next stage. Our goal is if we can generate enough data, biobanking, research, even some model organisms or iPS stem cell line, that it will make it easier for researchers to be able to take this and be able to develop, apply for grants and develop a research program from it, or to pull it together to do it in the more in-depth analysis. So in terms of the connecting, we have 3 different strategies that we're working on. One is through a patient registry to get all these patients and register not only in terms of what disease they have, but what's that genomic background. Secondly is sort of an online community for patients that communicate with each other with the same genetic changes. So, your favorite gene, you can collect all the people with the same genetic mutations, and maybe they can talk to each other. And lastly is patient-centric sharing to be able to sort of share that data. So the way we do it, the registry, we're currently working with Stanford Health to build a genomic registry. registry, not just based on diseases, but on their genetic background. We're actively in discussion with them on this. The second thing is we recently, Berkshire decided to join us, and it's an online community of 7,000 plus of the diseases, and we are in the middle of rolling out communities based on genes to allow patients to be able to connect to each other in this sort of social media way. And then lastly, we're developing what we're calling a rare disease commons, where patients are able to potentially, make open source or share their data in a very similar way that the PGP does it. And we look a lot to them. For rare disease patients, often privacy is of secondary concern, and they want to make it all public. And if they are the ones who make it public, nobody can stop them, right? But of course, we need to guide them to make them understand, and in a similar way that the PGP has very extensive training and testing to make sure people understand what they're getting into to be able to do that. So in short, we are sort of trying to build this ecosystem to be doing it, and this is, hopefully one effort of many to create this. And there are much larger efforts at the NIH, at academic institutions that are building this. But we hope to be one of the many voices to do that and take all these different aspects that we discussed and to be able to really help. So for Robert, actually, we've been able to connect him with some researchers at the NIH in Canada, and who are actively studying his cell line and doing functional studies for his genes. And we're excited that he not only gets a gene diagnosis, but hopefully one day would have substantive therapeutic options and to be able to help. And ultimately, Robert will be one of many, many patients, of the millions of patients that, more and more studies, not just sort of identifying the gene, but how do we really ultimately change sort of their quality of life and be able to help them. So thank you very much. That's a little bit a quick overview of what we're working on our GI. If you have any questions, happy to dialogue. And of course, as we see this as a large effort for the whole community, we have a large number of scientists who work with us, and we would love to sort of be in conversation. So here's my emails here, and feel free to connect with us, and hopefully we can together be able to help more patients together. Thank you very much. Any question? Okay. So I don't see any questions. If you have any further questions later, feel free to email me at my web address that's listed above. Thank you very much.